Para la próxima conferencia, las personas que deseen pueden pedir los equipos de traducción simultánea justo al lado de la entrada. La siguiente conferencia y última de este bloque de la mañana. For those of you joining us, here we're again to present this, the next and last speaker of this morning block. It's a great pleasure for me to introduce our next speaker today, Dr. Dushyantha Jayaweera. Dr. Jayaweera, known as Dr. J, has been a friend from the U.S. For a long time. Dr. Jayaweera ha sido profesor de medicina en la Facultad de Medicina Miller de la Universidad de Miami. Muchos de nosotros hemos tenido el placer de ser sus alumnos y rotar en el servicio de inmunología especial, como se llamaba en aquella época. Dr. Jayaweera es licenciado en medicina en Sri Lanka y oriundo de Sri Lanka. Ha realizado estudios de posgrado en el Reino Unido y en los Estados Unidos. Es investigador y especialista en VIH durante los últimos 20 años, o más de 20 años. Comenzó su carrera en medicina de VIH desde antes de la existencia de la terapia antirretroviral y ha desempeñado un papel muy activo en ensayos clínicos de antirretrovirales. Actualmente el doctor Jayaweera es el investigador principal de contacto del estudio de vacuna COVID-19 de Janssen, patrocinado por el National Institute of Health, el estudio de la vacuna antigripal mRNA de Pfizer y el estudio Active 6 patrocinado por los Instituto Nacional de los Estados Unidos y otros estudios de industria en materia de VIH, hepatitis C y COVID-19. Dr. J, welcome to Colombia, welcome to Bogota, and thanks again for being here.
Thank you, William, and thank you, Ernesto, for inviting me. And I have a lot of my colleagues on the audience, Hector. That was an excellent talk by the previous speaker. In fact, she stole half my talk. So anyway, it is what it is. But it's always repetition is better, so that you stick to what's really important. And I may start off with kind of simple. And because there's this translation, I'll go a little slow so that people can understand. So without further ado, I'll get started. So when Ernesto asked me to talk about PrEP and what's in the future, the previous speaker gave a lot of epidemiological data to make the case that PrEP is essential. Obviously, I have one more slide which just shows what exactly is happening at least in US, the HIV incidence and population among people over 13 years. So as you can see, if you look at the black population and the Hispanics, the number of people who are getting HIV or infected with HIV is much higher among this group, disproportionate to amount of people who are living. So, which brings to the point that we have to do something to prevent African Americans and Hispanic people living in the United States not getting the infection. So if I bring this up to Colombia, it's very similar situation where people who are vulnerable, who should get. But what reality happens in US is that the people who really need PrEP doesn't get PrEP because most of these people are uninsured or underinsured and don't have the resources. And so when people look at the United States and say, oh my God, they have everything. Yeah, sure, for a few people. But lots of people have absolutely nothing. So, so we are trying to change the paradigm. We are trying to make it different. But the reality is that, as Peter Piot once from the WHO said, United States has a big problem. We have a third world or a developing world inside the first world. And we have not truly recognized it and done something about it because we want to pretend that everything is good, which is not. So I'm going to start with something simple. This has been already mentioned about Truada. Why you are using Truada? All of you know that, but I just put it for completeness sake. I mean, people who are on high prevalence areas or high sexual activity in a risky area should get PrEP started. So, which means that, in Kensington, no, people are not using condoms or diagnosed of sexually transmitted diseases or using drugs or, exchange for sex for commodities or money or whatever, or people who are in jail, that they should be getting PrEP. So, I'll get to the problems of PrEP. So, if you look at that population, if you look at this population, they're the most vulnerable people. They are the people who don't have insurance. They are the people who don't have access to healthcare. And that's where the problem lies. That's where all these things lie. So we can come here for meetings and talk about this, but until the health system and the leadership and the governance change, nothing happens. In fact, there is a theory that everything we doctors do affects only about 10% of the health of the people. Because what really changes a person's life is the education, jobs, where they live, and what's available. That creates 90% of the health. 10% is what you and I do. So going from there, so obviously we have already discussed this, Truada is, everybody knows about Truada. It has a very safety profile, good safety profile. Drug interaction profile we know because it was approved in about 2002 or 3, and we have used it over and over again. Multiple studies have showed this, and it's just 1 tablet once a day. Now, I think the previous speaker alluded to some of the studies, and I don't want to go into details about each of these studies. And basically, if you take PROUD or IPAGE or Kaiser or all these studies, they fundamentally show that this works. This works. So, which means that everybody should be quite happy with this. But the question is, it is one thing for us to say that this is working. It's another thing for the person who is taking to ask, is this for me? Because lots of times, doctors are very good at telling people what to do. And it's the thing is that unless you really know what the patient is able to do, it is never going to work. So this is a question about, they ask our MSMs what the preferences are of type of HIV prevention. The total sample of MSM and the MSM not in relationship and the MSMs who are in relationships. I mean, most of this work has been done because that's the biggest group that are driving the HIV epidemic in the United States. But in Latin America, maybe it is slightly different with more heterosexual transmission. Like, for example, Africa is almost quite significantly heterosexual. So if you look at the total sample, the one that is most requested are the implant not visible. So you can see now how the world is changing. The people now don't want the pill, but they want an implant that is not visible. That's what they want. Then comes the next one, which is the implant visible or not visible. And then the pill takes a low ranking, although like, men who have a relationship with men who has a stable relationship, they are okay with the pill. So you can see the psychological aspect of it, because if you have a close relationship, then it's okay, you can disclose and you can take a pill. But if you're not in a relationship, then you're kind of, it's difficult. And also that, you don't know when you're going to have sex, so there are problems. The on-demand PrEP is not approved, although it is very effective. It's very effective. But so you get the picture that, so the population is asking for something different. So we have to do what the people want. I mean, that's one basic principle in medicine. I think in my last lecture yesterday, I said about that it's good doctors treat diseases, best doctors treat patients who have diseases. So which means you have to know what the patient lifestyle and everything is like. The second thing is that, once Voltaire said, perfect is the enemy of the good. So we had to be careful trying to look for the perfect, because we may destroy what is good. So we got PrEP, which is Druada. I mean, we may get, we are having the injectables. The implant could be the best, but it may not come in a hurry. Meanwhile, we will be infecting other people. So here is the requirement, but what the transgender people who are asking what they would like to do and what their responses are. So as you know, the transgender ones has the highest HIV transmission rate, the previous speaker already alluded to. So if you take the sexually active 100%, only 83% were aware of PrEP, but it didn't matter whether they were adults or youth, and then the ever taken PrEP 23. At the end of the day, only 8% of the transgender people were taking PrEP. So, and you can see, so the question is why? I mean, they are no different from all of us. I mean, MSMs and other MSMs are doing it. The reason is that the discrimination that is happening in healthcare systems and the total system that does not understand the lifestyle of what about the life of transgender. So that's one of the reasons what it is happening. So now the question is Truada or TAF better? And you know, how about TAF for PrEP? I mean, you all know about this TAF, you know, it moves very fast. You know, it's a prodrug, both are prodrugs of tenofovir. And then, of course, it has a less concentration in the blood and high concentration in lymphocytes, and it works better. So the Discover study, which was done to compare, it's a randomized non-inferior trial for FTAF for PrEP, basically comparing FTAF versus Truada. And the primary analysis of HIV prevention was done when 100% complete 48 weeks. And then it was followed up with open label. So what the study showed was this. So if you take the ones in blue, which is the FTAF, and the red is the tenofovir, you can see there's a clear difference in the effectiveness of preventing HIV, whether the relative risk were like 0.47 to 0.55, and it definitely met the non-inferiority margin. It didn't actually hit the superiority margin, but it clearly looks quite promising. So the advantage of FTAF was that the bone safety was much better. And you can see the statistical differences. And the renal safety was equally better. So, I mean, you know all this stuff, you know. I mean, I'm sure all of you have done doing HIV and you have been using TAF and you know all these different ratios. This is not something new. So what's important, the difference is that here you're going to give something to somebody who is not HIV infected, who are not having any problems, and saying, I want to prevent you from getting a disease. But by the way, it might affect your kidneys and bones. So this information is quite useful. So which medication should I prescribe for daily PrEP? I mean, obviously, FTAP seems to have an edge over tenofovir in MSM and transgender and heterosexuals and IV drug users. But for women, this has not been still approved, cisgender women, because the studies have not been properly performed. And of course, advantages are already mentioned. Now, the previous speaker spoke about HPT in 083 and 084. 083 was on MSMs and transgender. 084 was on cisgender women and the group that were susceptible. And fundamentally, it was kind of very similar design. People got either cabotegravir PO and Truada PO, and then after a brief period, they got these are placebo controlled. So either you get a placebo injection or placebo tablet, and you go up to I think it's about 48 weeks, and then they switch on to open label. So what the primary endpoint was 083 and 084 showed was that there was a clear difference. It's already been mentioned in the previous speaker that there was cabotegravir was better than Truada. So scientifically, that's good. Scientifically, it's good. Now, if you dig down into, so what happened to this? You see that there were 13 people in cabotegravir failed, and 4 people on HPT and 084 failed. So why did they fail? I mean, you're going to ask, like, because once you give an injection, you expect, for God's sake, it should work. You gave the patient the injection. So if you look at the individual cases, you can break them down to 4 groups. One are the people who got a baseline infection. The others ones were the infection after prolonged delay, because just because you have given infection, that doesn't mean that everybody comes for the injection on time. Things happen in life. When life goes on and some things happen, you just can't make the clinic appointment. So that could happen. The infection during old and leading phase could happen. Then the infection despite online cabotegravir dosing. So there were clearly things not going well with this study. And if you look at that green star, that's the HIV infection. So if you take the first 4 cases, these people were infected before they got into the study. So you can't help that. This has happened in other PrEP studies where, people come in and, they are infected, they don't know, and we don't test properly, so it gets there. The second group, infections after prolonged delay in cabotegravir dosing. So you can see that so if you have a gap, and of course the level starts dropping, then you can understand that. And the third group is the infection during oral leading phase. So that could be that, that they may have missed a couple of doses or something like that. And the final one is that the infections happen on-time cabotegravir dosing. So that is something that you cannot explain here, because why did they develop HIV while they were on-time dosing, adequate levels, and that is still a question that we are not sure. So this brought about an important change. This whole thing brought about an important change, the CDC, how they set the guidelines on how to do PrEP. I think the previous speaker talked about, the follow-up and all that, but it fundamentally changed. So here's the summary of the cases, So there are the baseline cases and there are the incident cases, which rolled out to about 16, combining 083 and 084. So if you dig down deep, so what Myron Cohen and the group from HVTN did was, they went and looked at the stored blood samples. They went back, every blood sample that from the day they tested positive as HIV, and went and did single genome sequencing of the virus over the previous couple of weeks and try to find out when did they actually get infected. And the results showed that they got infected many, many weeks ago, and we missed the bus. We missed the bus. Now, missing the bus is very serious because cabotegravir is an integrase inhibitor, and if you develop integrase resistance, now you lost a huge chunk of the treatment armamentarium. So as you can see, the first HIV positive 14 weeks before the first site positive. So this led to change in the guidelines, how we follow people on PrEP. So here is the delay of infection. The baseline infection was 4, incidence was 7. The median delay was 62 to 98 days. So one could think about this and say, we got to do something better than this. And mind you, these are clinical trials. These are highly well-regulated, well-conducted with great resources and kind of, highly motivated people. This happens. What would happen in real world? So these are things that we have to really be careful, okay? So, so here is comparing the cabotegravir versus tenofovir. I think this is an abstract from, I can't remember which study. And you can see that huge difference between the incident cases between Truvada and cabotegravir. So it's, playing the same point. So this I already mentioned that, the single genome sequencing gave us the idea that just doing a 4th generation Abbott assay to find out whether HIV positive or negative may not be the ideal for PrEP follow-up. So, which Again, then I would bring to another point about the weight gain. There are two things that with this HPT in 083 and 084 showed was, one was the injection site reactions. So most of these drugs that we are given injectables have injection site reactions. I mean, sometimes people get nodules, some people get, the redness and different things. The permanent discontinuation is about 2%, it's not that heavy. Any injection site reaction was about 32% for cabotegravir. And the weight change was not significant. There was a mild weight change, but that's not much significant compared to Truada. So again, Truada is one of the drugs that doesn't contribute to weight gain, so which is the why that there is a difference. So this led to CDC changing the guidelines, or I think the previous speaker tried to show this slide, but it wasn't showing very clearly. But this is basically saying that you have to do qualitative PCR on patients at every visit to see whether the persons are infected with HIV. So just doing an antigen antibody test is not good enough. You have to do qualitative PCR. Now, if you do quantitative PCR, you may miss it, because if somebody is infected and have a very low level of viremia and it doesn't get picked, I mean, you could have a problem. Now the question is, who on earth has resources to do a qualitative PCR in a clinic where you're running for the most under-resourced people? I started off by saying the people who needs PrEP are the most under-resourced, which means the place that provides support is also going to be under-resourced, because rich people get everything, poor people get, not everything. So as such, this is a huge challenge, even for the United States. How do you provide this problem, solve the problem? So if you had, say, for example, you see a paper, say, if you see oral PrEP person, you see every 3 months, we all know that. For the injectable, every 2 months. So when you see them, you have to send plasma for the antigen antibody assay, and also qualitative, they say qualitative or quantitative, so they are trying to be as easy as possible, because they know that lots of people don't have qualitative assays. And then if you have positive test, then that's it. And if everything is negative, you know that the patient doesn't have HIV. But if there's discordance between these two, then we have to repeat it again and see whether the patient has HIV. So these things are not simple. So this is one of the biggest problems with PrEP, because we have to get it going. So, and then the other issue was the PrEP STI, which was already mentioned. We see so many cases of STI with PrEP that is unbelievable. Multiple studies have shown this. And this was already shown, the effectiveness of PrEP in randomized clinical trials. You can see that partners PrEP, HPT in 083, 084, clearly shows that there was a difference in incidence, and this is a much better thing. So, okay. The CDC PrEP guidelines have here is given very clearly. Discuss PrEP for all sexually active people, prescribe to anyone who asks for it. That's very important. And then, of course, medication for PrEP. You can look at you can see, as you see, that is approved for treatment as PrEP. The drugs I mentioned. You have to do the HIV antigen antibody test. Then you may consider lipid monitoring. And then, of course, check the creatinine clearance. And, of course, do the qualitative and the quantitative PCR. So what's imaging PrEP? So people have tried tenofovir alphenamide as a Gates Foundation tried to do a sort of a long-acting injectable PrEP. That just didn't work. And after a long delay, they decided that they are not going to pursue this, because there are other options. Then, of course, dapivirine vaginal ring was mentioned. It's not considered for the approval. I know the previous speaker was quite optimistic about it, but the success rate of it's about 33 to 40%. So it's very low compared to 98 to 100% with other PrEP measures like cabotegravir. So I'm not sure that providing vaginal ring would be optimal for PrEP, because the problem is the microbiome. There are some multiple things in the vaginal microbiome which change depending on how your lifestyle is, and that will have an interaction with tenofovir. In fact, the tenofovir metabolism could be changed quite significantly based on the microbiome, and that is something that is really important. There was a whole paper published by one of our colleagues who went to Minnesota on this microbiome and vaginal microbiome prep. So the new ones are the islatravir. The islatravir is this nucleoside reverse transcriptase translocation inhibitor. And basically how it works is that I think I mentioned yesterday, it acts as a nucleoside analog, and also it has a chain terminator. So by both the activities, it'll block the transmission or replication of the virus. So this is being worked out for PrEP, but the problem is that it's on hold now because it causes a leukopenia. So as a result, islatravir trials are on hold. We are hoping that they will develop in the future, and there is a sister drug developed by Merck. So I hope that at some point it'll come out. And you can see that the islatravir blood levels and mucosal distribution is quite significantly high. So it's very good. I mean, this is a great drug. But the only problem is it causes leukopenia. So hopefully, if that can be settled, it'll be a good drug to follow. Then comes Lena Capovia. This is the first-in-class HIV capsid inhibitor. As you all know, I mentioned that yesterday, the capsid is the cover of the virus where the genomic material is kept. And when the virus enters, the capsid has to be disrupted. So that gets disrupted by Lena Capovia. And also when the capsid assembly, the Lena Capovia, the drug prevents the capsid assembly. So altogether, it's an amazingly powerful drug. And the beauty about this is that this can be given, it has a very long half-life, and it can be given every 6 months. So it's an amazing way to give PrEP if this can work. But unfortunately, this is also on hold because I was one of the investigators in one of the studies. It had an effect with it reacted with the glass in the borax silica glass, and the silica used to break into the lenacapavir solution, and it's now on hold. They are working on better glass than drug, because it's not the fault of the drug, but it's the fault of the glass. So, and the purpose study was to see whether lenacapavir compared to Truvada would work. So this is on hold. So the PrEP future, what is next? So the thing is, as I showed you a slide at the beginning where the people said that, we want something that nobody can see. You can stick it in and then keep it for 6 months and nobody can see. So either cabotegravir reformulation, double strength concentrations, you can have a microarray patch, you can put it and keep it. And then all the other thing is the cabotegravir implant. And also even Merck was looking at their islatravir implants, which they have been working. So there is a huge pipeline of drugs that are currently being worked, and we don't know how much of this, some of these things are very kind of a proof of concept studies, so we don't know which one will come to come to fusion. So as you can see that the present is what is available now, cabotegravir injections, and also PrEP, Truvada or Descovy. Emerging would be things like polymer ring, if they can work it out. The future would be obviously putting in an antibody-infused implant or implant which is either islatravir or lenacapavir, or the newer drugs that multiple companies, I think every pharmaceutical company is working on a drug that can be implantable, which can be done without stigma. Daily pill is not suitable because people don't like to take this pill every day. So with that, I want to thank the organizers for inviting me, and I thank my colleague Ernesto and William and all my friends who have invited me, and for the audience for listening, and I'm happy to take any questions. Thank you.
Muchas gracias, doctor G, por su conferencia.